Hi, my name is Gabriela Barrera. My group is group number 1, and we had the topic for flow cytometry. Flow cytometry is a technology method that is becoming very popular. Um, there's some examples listed as to what is being used for, and the main purpose of this method is to sort cells and separate them by size, shape, and proteins. So how does flow cytometry work? Flow cytometry is composed of 3 different things, which is the fluidics, the optics, and electronics. All of these 3 components make up the whole technology method. As you can see in this gray picture, it shows how it works, and you can see on these purple dots inside the fluid is the cells, and it's when it's called a heterogeneous mixture, meaning there's cells of all sizes and shapes. It goes down the sheath, and this laser will hit a single individual cell at a time, and it will do a forward scatter detector, meaning that it will tell you the size of the cell, and it's just based on how much light is reflected onto that detector that tells you the size of the cell. If there's a fluorescence tag like either protein, DNA, or RNA, then it will excite a light, and one of those lights will be of color, a fluorescent color, either green, orange, red. There's up to 6 different ones, as you can see on the electronics picture. And each one that's excited will send a signal to the processor, and it will show also on your computer results, your data. And on the results, you can see a scatter plot, and it shows like the size, and also the fluorescence color present. And the picture at the very bottom in the corner, the blue dark one, is how a flow cytometry machine actually looks like, especially when it's turned on and going through this whole process. You can see there's a red, green, and blue light, and I think they said this one had up to 6 paths, which is the ones you see here on electronics. So this is just a quick summary of how flow cytometry works. As you can see, where I circled in red and highlighted in yellow is where the fluid goes through, and it becomes hydrodynamically focused, which means that the cells are getting lined up to go down one by one. The air and other gases pressurize the fluid, and that's what controls the rate at which each cell is injected or released down, and the laser hits it. As explained previously, the light is reflected onto a forward scatter detector, and the size is being shown on the scatter plot and histograms. If the cell contains a fluorescent tag, then it will excite a fluorescent light, and everything that is collected will be shown on data scatter plots or histograms. So this image is a representation of the results obtained in a research that was only mentioned in the paper that I read. I did go to them to their actual research paper as a reference they had, but it was a little complicated to understand. But from my understanding and from the paper I read, this is a plate, an agar plate that was made straight from a flow cytometry machine method. And each line, the red, it's a DS red, and the green is called a GFP. And these two contain a protein that they were trying to see if it was present or not in the plasmids that had DNA inserts. And the purpose of this was to get results that would help them clone the E. coli bacteria. As for my questions, I don't have any specific questions. It was pretty easy to understand the way this method works and what was the main purpose of it. My only question is, how did people come up with this amazing technology that is becoming very useful and helpful in the medical field. Also, I wonder if one day this technology could be used for all kinds of diseases, not just anything that's related to blood cells disorders. And here are my references.